I'm going to be talking about how we're applying integrated genomics to precision care or precision medicine for bladder cancer. And that's a big initiative at our cancer center. And let's see. Okay, and I just wanted to get this kind of up front. It's a huge team effort that we have here to do this. And so we have our genomic shared resource, and Ryan Davis and Stephanie Liu are the main people doing a lot of the work. But then we have a big oncology group that works on the mouse models and clinical translation of everything we're doing. And a big kind of the driver of a lot of this work is our Cancer Center Director, Ray White, and this is also being done in partnership with the Jackson Laboratory. And also, I should also mention that Dr. Chong Pan is pretty much the leader of our bladder cancer initiative. And so I'll just give a little brief background of what bladder cancer is. And according to the American Cancer Society estimates for 2015, there'll be about 74,000 new cases diagnosed, and unfortunately 16,000 people will lose their lives to this cancer. It's generally referred to as transitional cell carcinoma, and that's the most common type of urinary bladder cancer. It's also referred to as urothelial carcinoma because it mainly occurs in the urothelium or urothelial layer, which is the inner layer of the bladder, and this is where the bladder is. And generally, there's two main groups or types of bladder cancers. There's superficial, which accounts for 70 to 80% of the cancers, very high and very good survival rates. And but then the really problem tumors are the invasive, what's generally called muscle-invasive bladder cancer, MIBC. And that's when you have the tumors going from this transitional epithelium and invading into the musculature of the bladder. And you have all these different stages or staging of a cancer based on the amount of invasiveness. And when you have a 96% 5-year survival rate in superficial cancers, this drops to 69%, to 34%, and to 6% for metastatic muscle-invasive bladder cancer. And this is the real problem. Treatment for this type of cancer usually involves removal of the prostate with a radical, of the bladder with a radical cystectomy, and very toxic platinum-based chemotherapy, which is usually cisplatin or carboplatin, plus or minus gemcitabine. And really the real critical clinical unmet need is that there's been no significant improvement in overall survival and prognosis for the last 30 years. So we really need to select better first-line therapies and salvage chemotherapies, and try to select better targets for treatment. And as far as precision medicine, I think a lot of you are familiar with it. It's a lot of times referred to as personalized cancer therapy, and where we're basically trying to tailor the therapy to a patient's specific needs by matching the treatment strategy to molecular features specific for their cancer. And this has been standard of care for several types of cancer with well-defined aberrations, specifically in ERB2, EGFR, KRAS, where you can run the Scorpion-Hardens assay to see if there's mutations that are going to be clinically informative for therapies. A lot of this has been revolutionized by next-generation sequencing, where you can get rapid and comprehensive molecular characterization of tumors, either with Whole-Exome sequencing approaches for gene mutations and structural variants, and gene expression with RNA sequencing. But even with all this, a significant challenge is to identify the most efficacious target. And just to kind of give you a brief background on what the genomic features of bladder cancer are, a lot of this has been learned from these very large multi-cancer center analyses of bladder cancers that have been performed through the Cancer Genome Atlas Research Network. BGI had a nice publication as well. And a lot of really fantastic analysis has been provided through Ingenuity, the group doing that. And one thing that's come from all of these analyses is that bladder cancer has one of the highest mutation frequencies amongst all cancers. It's a solid tumor, and it's up there with lung and melanoma and the kind of carcinogen-induced tumors. There's about 10 mutations every megabase. And from all this work, there's been about 250 driver or significantly mutated genes identified. And a lot of them, was probably to be expected, are involved with tumor suppression, chromosome dynamics, transcriptional regulation, signal transduction. And there's another number of kind of usual suspects that have now been found, and particularly like the FGFR3 fusions with like TAC3. And typically, 50% of the cases, you'll have a mutation in one of the gatekeeper genes. And some of the key pathways for targeting these cancers that have come out of PI3K pathway, receptor tyrosine kinase pathways, which includes FGFR3. And so that brings us to our study, and there's multiple goals. One of the main goals was, or overall goal, was to develop and characterize a platform of bladder cancer patient-derived xenografts that can be used for precision medicine, that it'll enable us to do in vivo testing of therapeutics. And some of these sub or specific goals are, from where I stand, is to molecularly characterize these PDXs using integrative genomics. And so we've conducted whole-exome sequencing, RNA-Seq, SNP genotyping, and the microRNA expression profiling. And following from that, one thing we feel is important is to evaluate the integrity of these patient-derived xenografts as models of cancer. And then one thing that I'm interested in is developing and evaluating target selection strategies that might lead to improved responses from personalized cancer therapy. And so just an overview of our study design. We have basically 8 tumor models, 8 PDX tumors established from 8 individual patients. And this is the clinical features, usually older patients, various stages. These are all muscle-invasive bladder cancers. Over half of them had no prior treatment before being put into the mice, and 3 of the patients did have cisplatin-based or carboplatin-based therapy. And the advantage of this in establishing models is that, we need more tumor models. And what they also provide is a living tumor panel that has defined clinical features and represents individuals with diversity. So it's not like a clonal cell line that we're always working with. And basically, to establish these, the patient comes in, and after either a radical cystectomy or a biopsy, the patient tumor is actually sent over to Jackson Labs West over here in Sacramento and put into mice. And then they're grown up to their initial passage, which is P0, and then it can be continually passaged. And then what we'll do is the genomic profiling from the original patient tumor and from the xenograft. And I think I spoke about what the profiling was. And then following that is a lot of bioinformatic analysis, as you know. And so this is just an overview of the whole genomics and bioinformatics analysis workflow. And so we did whole-exome sequencing using an adjuvant SureSelect target enrichment, all-exome panel, and with Illumina sequencing. And then we basically just run Burroughs-Wheeler aligner genome analysis toolkit pipeline And after getting the variants and genotypes, through running genome analysis toolkit, then we put it into Ingenuity Variant Analysis to identify causal variants, prioritize the variants, and also help with the biological and functional interpretation. And so going to the first goal, we wanted to validate the genomic integrity of these tumors. And so basically we got the VCF files from the patient after analysis of the patient tumor, or that first passage in the mouse, which is the P0. And basically we just did took our FASTQ formatted data, obtained variant calls, and then put it right into variant analysis. And this was the filter cascade that we set up. I think we just modified the default. One of the things I found that was really, and here's the specifics of each of the filters. We basically, had it select for the upstream pipeline filtering that had to pass. We wanted, actually the call quality I actually have a different number there. I think it should be greater than 50. But we generally, since these are cancers, we want a read depth of greater than 50. We try to hit 100, 100x coverage. And for allele fractions, we'll start low at 20% at least. And then we want high quality calls, and then we do various, we'll set up the filters according to various regions of the gene or genome that we want included or excluded. So we really weren't that interested in UTRs or enhancers, so we uncheck that or ask them to exclude it. We're primarily interested in genes that were variants that were going to be present in the exonic regions, and that we will be a little bit easier to interpret in regards to function. And that's right here, where we're basically which there's the protein coding effects or phenotypes, either a missense mutation or something that might be pathogenic. And also we're looking for cancer-associated pathways, and also variants that are found in Cosmic or TCGA database that's greater than 0.01% frequency. And so this, using Ingenuity Variant Analysis made it extremely easy to do this. I think one of the advantages of using it is that, well, we can set these things up on our computer. The nice thing is that all the databases are up to date, and we can actually output the filter cascade, and it'll provide all the content versions for us. So that's particularly advantageous when we're going to write a paper. And then in terms of what the results gave, basically after applying all these filters, the patient tumor had 233 somatic mutations, and after passaging in mice, the mouse tumor had 208 of those mutations. And so integrity is validated, and we break it down. Here is basically where the somatic mutations we're referring to here are either missense or insertion deletions. And these were all the variants that were found. But generally, when we come over here, you can see the percentage of variants in common. And so usually just looking at all variants across the board, it's over 80%, but for somatic mutations, it's about 90% conservation in the mouse. So they are decent models of human cancer. And so now what we wanted to do is characterize our whole panel. And this is where the IVA was very, very handy. The first approach we took was, one of the interests is, of course, bladder cancer. So we focus on genes that were either known to be bladder cancer drivers or significantly mutated genes in bladder cancer from TCGA or sourced from Qiagen. One of the considerations that we had to make was that all these analyses were done with tumor sample only, and so normal tissue was not included. So in order to address this, all the variants should have confirmed status of somatic mutation in cosmic or TCGA, and we want to remove variants associated with human genetic diversity at a frequency of greater than 3%. And IDEA was very helpful for this, and that it kind of consolidates all these databases together for us in that interface. And we want to remove variants potentially in dbSNP, but that a lot of times can be too stringent. So we evaluate that kind of at the end. And here is our filter setup, filter cascade setup, which basically is similar to what we had set up for comparing the mice. But what we end up doing is we kind of apply our hard filters or standard technical filters. We then filter out the common, the variants that are common. And then we start to select for the variants that are going to be of phenotypic or pathologic impact. So we'll look for deleterious mutations, either missense or frame shift. And then we go down to looking at what's in the cosmic or TCGA database. And then we also have our biological context, so where we could upload our, in addition to using the filter built into IVA, we upload our own set of genes into the biological context for genes that we've been interested in through TCGA and Ingenuity. And then a couple of the other things we keep in mind are the allele frequency. For certain targeting applications, I think it might help to have an allele frequency that's somewhat high, so we're targeting a higher proportion of the tumor. And here's just a snapshot of the results when we're doing bladder cancer focused analysis. And here's the screenshot from the Ingenuity Variant Analysis. And in brief, we got 25 non-synonymous variants occurring in 21 genes. So this is the whole data set. And interestingly, there's a potentially activating mutation in PI3K in the example BLO-269. Most of the mutations result in a loss of function, but with having uncertain significance, and 75% of mutations termed pathogenic were occurring in tumor suppressor genes like CDKN2A or P16 and P53, and also yielded a number of actionable targets. Which is exciting. And so so when you look at the screenshot, and these are the results that you get at the website. It's very easy to understand and use, and it shows you what the transcript and protein variant is, and what samples it occurred in, and whether or not they are inactivating or neutral. And also, here's the activating one, which is the PI3K mutation. So a very user-friendly interface. And then this is just kind of, after looking at all the data, we summarized it in this table, and we relaxed a couple things, but basically we got 71 mutations across the whole panel of 8 tumors. And we can see there's a total of 43 different genes, and really not a lot of mutations in these bladder cancer specific genes, in each of the tumors, which is expected out of about 240. But we break it down to what was known as a TCGA driver or BGI discovered driver. And so now we move on to kind of a more comprehensive analysis. And this is more of a pan-cancer approach. And the reason we want to do this is it increases the likelihood of identifying increased numbers of drivers and potential therapeutic targets. And in addition to individual genes, we have a better chance at pathway analysis revealing more robust targets. And so here's the filter cascade that we set up. We kind of moved down the biological context, which has the bladder cancer specific genes, and now we also uploaded our filter for therapeutic targets. And we actually played around a lot with this because depending upon where you finally make your selection, we broke up the predicted deleterious filter into two different filters, where we separated the pathogenic from just the amino acid affecting mutations. And so we generally like the results that we got here at the cancer driver variants. And there was more to work with. When we started to impose the filter to remove Stevie SNP variants, we get a lot less. And so this seemed like kind of a good sweet spot to work with for kind of pathway analysis, and also when we imposed therapeutic targets, which we actually moved up. And so this picture you get from Ingenuity Variant Analysis when you just hit summary in the window. And so there's over 5,000 variants across the 8 tumors. And what's remarkable is that there's not a lot of variants that are termed pathogenic or likely pathogenic. And here's the breakdown of them. This BL479 had most of these pathogenic mutations, but they're all pretty evenly distributed. 429 has, in general, very few mutations compared to the others. And so one thing we were able to do very easily in variant analysis was basically just use this pathway tool to perform pathway analysis, and it returns a number of hits for enriched pathways or pathways with genes in them, and the protein ubiquitination pathway was one of them. It was at 13 genes that were segregating to that pathway with 15 variants, and it was in 7 of the cases. So it seems like it might be something maybe universally aberrant in these tumors. And so these are all the genes that were found to be mutated and also in the pathway. And what was interesting is the USP6, which is ubiquitin-specific protease, is actually has an activating mutation. And what's interesting is, these are all reported to be in cosmos, so there are no somatic mutations. And so I think we can, I think in a way it's validated that it might be, significant to the cancer biology, and it is indeed targetable. And then here's just a snapshot of the pathway figure that we get from IVA. And so DUB is actually can be USP6. I think this is generic for ubiquitin protease. But it's nice that the pathway figure indicates where in the ubiquitin pathway of activation, and you also have a couple of genes which are have mutations that inactivate them as well. But if this pathway is activated in some way, it's a good candidate for proteasomal inhibitors. And then the final part of the talk is where we're beginning to just come up with rule-based or heuristic selection of therapeutic targets based on integrated analysis of whole-exome sequencing and RNA-Seq data. And I'll just briefly go over this. So basically we have a number of filters, approaches that we want to use where to filter through the data, and ranging from technical filtering to filtering the variant types, that we discussed, like missense and indels. And what becomes important is the functional filtering, where we want to find actionable genes or actionable mutations. So we want to identify variants that have functional impact, clinical relevance, might be present in pathway nodes, and we also want to impose certain criteria and integrate RNA-Seq data so that we know that the gene is actually expressed. And when possible, we would like to just integrate copy number data as well. And then also include allele frequency as one of the criteria, which gives us an indication of tumor heterogeneity, and also, what proportion of the tumor you're targeting. And this just gives kind of an overview of when we took 4 of the tumors, 269, 293, etc., and took all the variants and put them through the filter cascade we set up to kind of go to select for targets. And when you go through the different filtering modes, you can see that, of course, the number of variants decreases. And then, while we do get a number of actionable genes in each tumor at the end of all of this, say for 269, there'll be 90 actionable genes. When you now say, okay, what genes are actually expressed at greater than 10 FPKM, which is a low to moderate amount of expression, that 2/3 of the number of genes drops out. And then when you select for therapeutic targets, it's even less. And then finally, when you have therapeutic targets that are expressed, you have a lower number of genes, but these might represent your best targets. And so I think I don't have, I'm missing one slide, but basically we decided to run this on one sample, so variant analysis on one sample, and particularly BL269. And when the results of that gave us about 15 genes, and that had variants in it. One of the variants was this PIK3CA mutation, which was the H1047R mutation. And so Julie was kind enough to run this through the Qiagen Clinical Insight tool, and this platform provides an amazing amount of information that's I think valuable to any oncologist or clinician. And so, this is the kind of data we're always getting from GATK analysis, where we'll have the transcript variant and then the protein variant. But then what's nice is it consolidates all the information, and there's a nice report about the somatic frequency and cosmic. It shows that it's not in the population. It tells me how many reads I had in the sample. So it's 115 reads and a allele fraction of 50%. So 50 reads are, at least 50 reads are accounting for this mutation. And it tells you what the gene prevalence in cosmic is, and also that this is a somewhat frequent variant, and so it becomes more clinically relevant as well, and that it is a gain of function. And of course, the important thing at the end of all this is that there are clinical trials available for agents that do target an activated PI3K. And it's important that this variant, or this program tells you that it's activated, because it seems that the PI3K mutations that confer activation also are the most sensitive to these PI3K inhibitors. And I think the next page is just another view of the information you get. It shows that it is a gain of activity mutation, and all of this clinical information as well from ClinVar and Cosmic. and then the distribution of the mutations through all the known tumors that it's been found in, and the genotype. And so, you know, this is the kind of information that I think is extremely valuable for personalized medicine. And so there, I think I come to the end of the talk. And I think, yeah, so I'll just discuss this first as far as some of the key findings. We really need to find the Achilles heel of these tumors, understand what's driving the tumor and what will result in the tumor's death. And so we just don't necessarily want to, you know, targeting the drivers don't necessarily induce death, such as BRAF doesn't necessarily induce death of melanomas all the time. And what we think also is that an integrative genomic approach might yield effective target selection, and so considering genomic variants with expression. And I don't know if I mentioned this when I was going through it, but the way we did this was that we were able to upload the RNA-Seq expression data into Ingenuity Variant Analysis, so we can look at those together. And then that brings me to just some of the advantages of using variant analysis. It makes filtering extremely easy, and also gives you a lot of flexibility in your analysis. It makes it truly rapid. I really like that you can export the analysis steps and results. So there's transparency and reproducibility in the reporting. It takes standard text or VTF files, and then I mentioned transparency. And I really appreciate that the databases within the tool are comprehensive, and that the content versions are recorded and always seem to be updated. And it was very helpful to be able to upload custom annotation and content. I think this is an extremely powerful feature for customizing the tool to your own use.